This is Suganti, Assistant Professor, Department of Biotechnology, Vistas, Chennai. In today's class, we are going to see in detail about post-transcriptional modification. Post-transcriptional modification is nothing but pre-mRNA processing. So this post-transcriptional modification will take place only in the eukaryotic cell. So this is nothing but it's a set of biological process common to the most eukaryotic cell by which an RNA primary transcript is chemically altered following the transcription. So the nothing but this after the transcription process, the primary RNA transcript is undergoing some chemical changes like 5' capping and 3' poly A tailing and splicing mechanism to produce the mature and functional RNA molecule. So once this post-transcription modification is completed, it will be the mature mRNA molecule is exported to the nucleus and enter into the cytoplasm for the further translation mechanism. So in this picture, you can clearly see the prokaryotic transcription and translation mechanism. In prokaryote, the transcription and translation is coupled, but in the eukaryotic cell, the transcription and post-transcriptional mechanism, like the processing of mRNA, is takes place in the nucleus. Then after the post-transcriptional modification, the matured mRNA are transported into the cytoplasm for the translation process. So the primary mRNA must be processed to produce the mature mRNA active form. So it takes place in the nucleus, but this process only takes place in the eukaryotic cell. So the mRNA processing include there are three steps like 5' capping, 3' poly A tailing, and splicing. Capping is nothing but addition of the artificial guanosine molecule at the 5' end of the mRNA molecule, and polyadenylation we know addition of the poly A tail, that is addition of the adenine nucleotide at the 3' end of the mRNA molecule. Then splicing is nothing but removal of the intervening sequence like in trans in the mRNA sequence. First, we will see in detail about the addition of 5' capping. Here, the modified 7-methyl guanosine, it will add to the 5' end of the mRNA. It will so for what this 5 capping and 3' tailing process will take place means it will protect the mRNA from the ribonuclease activity. So in naturally the nucleases are the enzyme which is present in all where like in cell, inside the cell or outside the cell. So it will easily degrade the DNA and RNA. So to protect the mRNA from the ribonuclease activity, this 5' capping and 3' polyadenylation is takes place. This 5' capping, that is the addition of the 7-methyl guanosine, it will be added after the first 20 to 30 nucleotides of the mRNA. So how this 5' capping is done means by using the unusual 5' triphosphate linkage. Through this linkage, the 7-methyl guanosine, the modified guanosine, will be attached to the 5' end of the mRNA. So that's all about the 5' addition of the 7-methyl guanosine. The next one is splicing. Okay, splicing is nothing but removal of introns from the primary transcript and joining of the exon. We all know that introns are non-coding regions are present in the mRNA. So this introns does not play any role in the coding of the protein. So this introns must be removed before the translation mechanism, and the coding sequences called as exons must be joined, and automatically the mature RNA will contain the exons, not the introns. This is the very important mechanism, RNA splicing. So the how the splicing is done means it will be mediated by the large ribonucleoprotein. So this is a complex like spliceosome. This spliceosome contains a certain uracil-rich small nuclear RNPs and associated with protein, and it will form a spliceosome. And this spliceosome, it will come and recognize the 5' splice site and the branch site, and it will bring the site together. So the mechanism is simple, the removal of introns. So but here we must know how the spliceosome involved in the recognition and bringing the site together, and how it will cut the introns like. So here I have given the detailed notes about the splicing process. So in this picture, we can see the assembly of the spliceosome in the mRNA to remove the introns. Here you can see the SNRPs and four subunits will come and assemble. So this complex is known as a spliceosome. So it will automatically remove the introns and will join the exons. But you must know the cutting site and removing site. The next one is poly A tailing. At the 3' end of the mature mRNA, the around 80 to 200 adenine nucleotides are added. So for what this 3' poly A tailing will takes place means it will takes place to safeguard the mRNA, like to protect the mRNA from the nucleus activity. And also this acts as a binding site for specific protein that protects the mRNA from enzymatic degradation. So this is the picture representation. In this you can see the poly A tailing of the mRNA molecule. So at the end of the post-transcriptional modification, the mature mRNA, it will contain the 5' cap and 5' UTR. UTR is nothing but untranslated region. That means in this region there is no translation process occur. Then next one is start codon. It will start with AUG. Then the coding sequence of the gene. Then stop codon. It may be a UAA, UAG, or UGA. Then next is 3' untranslated region. So the gene, it must be protected by 5' UTR and 3' UTR. And then at the end, the poly A tailing. Okay, poly A tailing is present at the end of the mature mRNA, 3' end of the mRNA. So here I have given the definition for introns and exons. So introns are non-coding segments of the DNA within a gene that are transcribed into RNA but are removed during the process of RNA splicing before the final mRNA molecule is translated into the protein. So the introns are nothing but it's a non-coding segment. That means the regions of the intron does not code for any amino acids or for translation mechanism. But in the segment of exons are the coding segments are present in the exon region. And I have mentioned some terms used in the post-transcriptional modification. 3' splice site and what is 5' capping and what is 5' splice site. And here also I have given about what is exon, what is intron, and about the guanyl transferase. These are all the terms used to understand the detailed mechanism of the post-transcriptional modification. So after this, the mature mRNA will be transported into the cytoplasm for the process of translation mechanism.